Today we're going to be talking about the respiration lab. The respiration lab we're going to be doing is we're going to be using plants. Now remember, plants also do respiration, especially when they're very young, when they're seeds. As seeds, they've got an energy storages in there, which is a group of sugars, proteins, and lipids that they're going to use to be able to develop the rest of the plant. Now, if we're going to measure cellular respiration, if you recall, you're going to have to take a look at the waste products, which are carbon dioxide and water. Now with that, water would be pretty hard to measure, but carbon dioxide is a gas which comes out and it can get measured by this particular guy. This is a CO2 monitor and it's wireless. And it's part of what's called SparkView. You guys have probably used SparkView in the past. So what's going to happen is we're going to look at the different conditions and we're going to place this on top, attached to the SparkView, and then look at the CO2 that is being evolved over time. Now with this, I will mention there's going to be different conditions. And you guys have already talked about these, so I'm just introducing them to you. So what are the conditions going to be? We need a glass beads. And we need ungerminated mung beads. Now one thing to think about is why would we do glass beads and ungerminated mung beads? Interesting sort of thing. Mung beans germinate really pretty quickly. So this particular one, you can see in there that those particular ones, there are already roots that are showing up and that sort of thing. And this is after 2 days. This is actually after 5 days. We're going to be doing 4 days actually in the lab. This is done a day later. And so these, as you can tell, man, there's a lot of roots and they're actually developing leaves. And then another condition is going to be dark. Notice it is in aluminum foil to block any light. And it's going the same amount of days as this guy was. So glass beads, ungerminated, regular room temperature, 20 degrees Celsius. Then we're also going to run it 2 degrees Celsius, 37 degrees Celsius, and 60 degrees Celsius. In addition, we have one that's been in the dark, 20 degrees Celsius, and another one which is been out for like 5 days. So that's what we're going to do. We're going to take it, measure the CO2 concentrations. I'm going to give you the graphs, and then you're going to have to figure out what's going on with the respiration and why we're getting the rates that we're getting.